because I'm now presenting the German Plant Phenotyping Network, the DPPN. The DPPN is a project which is now actually coming close to its end. We actually have just extended the DPPN project funding until end of next year to install big installations. But otherwise, the DPPN is mostly now finished because it was a project and is a project to develop infrastructure for as a national plant phenotyping network within Germany. The mission was to develop and install infrastructures in phenotyping science, innovative concepts, specifically the question of lab to field and novel technologies. And we also had the mission in there to talk to the stakeholders, specifically academia, breeders, and industry in various means. The consortium in the DPPN is between Jülich, IPK, and Helmholtz Center in Munich. You already heard yesterday from Yogi about their activity, or some activities they have in Munich. IPK, you already saw in the previous presentation that there are facilities also available, and in Jülich you also saw some activities there. The reason why we set it up in that consortium was that we have existing experience in plant phenotyping in these three groups. They are complementary with respect to a science background. So we really like to put phenotyping activities into infrastructures into a specific research background, because phenotyping as a tool needs to be implemented into certain into a certain scientific background. The third very important point in putting this into these three institutions is that there has been in the past and will be in the future institutional funding from the research organizations running these phenotyping platforms also for long term. So one of the essential parts in the grant agreement with the Ministry of Science and Education in Germany was that the three institutions agreed that after finishing this project, for at least 3 more years, we will maintain the operational activities of the infrastructures which we have implemented through DPPN. What is there already? As I said, there is already an integration into institutional funded aspects. The LambdaTech facilities in Gardersleben, there we have tomographic systems, MR heads in Jülich. There are simulation systems. Yogi showed yesterday some of them. We also have field facilities, which we partly are using there are field facilities in Gardersleben. There are also field facilities which we use in Jülich together with the University of Bonn, where we have a very intensive equipment sites there, in which we are also bringing into this German Plant Phenotyping Network. During this DPPN project, on top of the 38 million which was invested from the Ministry of Science, the institutions themselves put a lot of money into infrastructures, building infrastructures. So this is this system here. You see 3 stories high. This is essentially one big climate chamber, which has something like something like 300-400 square meters of space inside, with 4 compartments which can be climatized differently. That's an installation in Gardersleben, which has just been opened. This one is the large greenhouse which we have built in Jülich. This compartment here has 800 square meters where we will install rhizotrons. And this is the greenhouse in Munich, which will specifically set up with rhizotron systems, which are able to run also plant pathology background. So these infrastructures, this housing, came from the institutions themselves and was not part of the project itself, but was obviously a basis for development. Don't look into details. That's the network structure of the project. Altogether, we had 54 work packages shared with different institutions, which are interlinked, as you see with the networks here, with the arrows here. All of them have been, most of them have been now actually finished. We are in, I think it's 8, something like that left, which are running. This is mainly those which are building up the large-scale infrastructure now as the output of the prototype development which we had before. We have a number of modules in there. For example, there's one which is DPPN seed. Just 2 examples for that. We have established in Gardersleben a time domain NMR system for seed analysis, which is able to look into individual seeds, to look into structure as well as function, as well as composition of the seeds over time, using NMR technology, and you're looking at the dynamics of seed development and seed filling. Another system which we have in the seed background is the so-called Phenoseeder, which is a single seed robot, which is putting single seeds into different measurement modes, from the scale of Arabidopsis, which you can't see here, which you can't see either if you're in the lab, to bigger seeds, and you can measure individual seeds in large throughput, and then do this kind of correlation issues here between the different measurement modes, reaching from individual seed weight, shape, density, all of these kind of things, color, these kind of things can be measured there, and we can do this at a high throughput using several thousand seeds per day. The big platforms, the high throughput platforms, which are one of the targets to be established within DPPN, are on the one hand a root platform and a shoot platform, or root platforms and shoot platforms for architecture and physiology, and then now we are integrating them into integrated root shoot phenotyping platforms. There are field platforms, again, for architecture and physiology on the stand level, for field phenotyping platform, as well as a special system to simulate future environments with the breed phase, and I guess that Onno will talk about this tomorrow. And there is a patho node, which is looking about biotic, so not only pathological, but also mutualistic and beneficial microbe interactions between plants and the microbes, from cells to tissues. The volatile emission platform you yesterday already have been introduced to by Yogi. We also have set up a resource with respect to material, which is a collection of genotypes which are very widely varying in their habitus in order to, from different species, but also within the species having that very wide diversity of habitus. The reason for that was that we wanted to use this material, have used this material to see how wide our platforms can actually be used with respect to variability of the structure. So it's about validation of non-invasive sensors and enabling multiplication, that the multiplication is existing, so we have the possibility to also spin this out to others in some limitations. The reference collections have been analyzed and controlled, semi-controlled in field systems, so that that's part of the aspect which I mentioned before, the lab to field transfer. So we know for these genotypes with a certain background as well, we know how they're behaving in controlled environment and semi-controlled environment in the field conditions. Another aspect which we have set up is the deep phenotyping parts. You yesterday have seen the CT work in Nottingham. We are mainly focusing at the moment on NMR and PET. NMR to use, we have 2 NMR systems running at the moment of different stamps and different size, and we're combining them with the positron emission tomography. The NMR mainly used for structural analysis as well as water relations, while the Pflanzen and the PET is used for carbon transport and carbon activities, or transport processes and allocation processes. We combine them with root models. Tony already yesterday introduced SimRoot. There's the open SimRoot consortium now between Penn State, Nottingham, and Jülich, where we provide this model also as an open source model for people to continue developing that. That's a structure function model, and we are feeding the non-invasive data into the 3D model, which is structure function. Some examples for that, what you can do with that. So you can see at different time scales how, for example, roots are developing in soil. So here you can see a root, how it's growing in soil over days. This is quantification. This you can also do in here. This is a quantitative analysis with respect to root diameter. So each of the colors has different root diameters. And then you can overlay the carbon flow, which is the red stuff here. So that's the C11 distribution with the structure. For example, also seeing nodules, how they are developing and how they are active with respect to current. That's the deep phenotyping in roots. We also have high-throughput phenotyping and are continuing to build on that. For root architecture and physiology, we have developed a number of different screen systems now. A lot of metal in here, which we are actually now getting rid of in the newer system. So have a very flexible systems in order to have rhizotron-based root systems. There are a number of different systems now, which, sorry to go back to here, which are now also which we also now spin out to all 3 locations. So there will be a rhizotron system in Jülich, there will be a rhizotron system as well as in Gallersleben, available with different focus. In Jülich we'll have a very big, very large one with a capacity up to 1500 to 1600 rhizotrons, large scale in that greenhouse which I showed you before, multi-trait analysis, and we will finish that until end of 2018. This also includes the whole pipeline from setting up the rhizotrons with packaging. So if you have 1600 rhizotrons, you don't want to pack them by hand anymore. And this is a whole semi-automated rhizotron filling and compaction facility. And on the other hand, we also have a lot of destructive sampling there, which we take care, including in the end, which is not on this picture, the washing of the rhizotrons, because you also don't want to wash 1600 rhizotrons by hand anymore. There's also a lot of substrate handling, pipeline handling, so this is also things which you have to think about early on if you build this kind of large scale through. On the shoot side, we also have a number of different infrastructures existing or technologies existing. On gas exchange, we have on photosynthesis, we have different fluorescent systems in place, we have gas exchange in place. We have water relations with different backgrounds, not all of them are on the screen here, including crazy new things like microwave analysis, where we can measure quick things, or dynamic thermography. Then we have growth traits with different aspects and also structures. So there's a whole portfolio of methods which are available now in the platforms to measure key parameters for shoot parameters, and they are implemented into higher throughput systems. For example, the system in Gallersleben, which is a LambdaTech-based system, plus in the future they will also have bigger systems based on the rhizotrons, but also on containers, which are also holding large plants. We also have systems which are automatically weighing and watering units, not only in the moving field systems, but also in stable table systems where we can water and weigh individual pots and therefore get information from water relations of them. In the field, we have also been active a bit, not as much from the DPPN, but rather from other sources. There we have key traits looking into photosynthesis, or key traits are canopy photosynthesis, spectral information, canopy structures, and also the breeding plot design, so the combination of the breeding plot designs. In many cases, we are actually integrating across many of this data in order to also do data assimilation and try to use this information with respect to, for example, 3D structure combining with spectral information gives additional information that is actually needed in order to interpret the data properly. We are having a full sequence here from traits to sensors to positioning systems to specific experiments like the face, for example, or the common experiment where we test the different genotypes, to environmental sensors at various levels, and this is installed and running now with respect to the infrastructure, and it's obviously also included in specific experiments also at the moment, which are also accessible through DPPN 2020, part of them, but also beyond that. Within the field structure in IPK, they have an additional system which they set up based on a hyperspectral imaging system on a phenomobile. Essentially here they are trying to do some root phenotyping in the field. Essentially this works by putting nutrient bands in different depths in the field and analyzing the color, the reflectance of the crop based on when they reach nutrients. And therefore you can indirectly see how effective the root system actually is. So that's one example how you can use this kind of systems in a combination with certain experimental setup. As I said, we also have the common experiment running now. I think it's the fourth year now, fourth or fourth year, where we are growing wheat, barley, corn, and rapeseed, different genotypes in the field, in a plot design which we are using together for, on the one hand, with breeders, on the other hand, with technology providers from different background. We are going there from the in-house closed environment through bigger rhizotrons, through these kind of container systems, which we also have installed in Klein Altendorf, close to Bonn, which is an automated system, which is where you have these containers, each of them being a cubic meter roughly of soil, where we have assemblies of plants, and then we put the same genotypes out in the field in order to do this lab-to-field transfer. We're also doing meta-analysis to look into the literature there, and one of them is cited here, where we mainly address the question of how this translation of trait analysis from single plants to the field, from different structures, different indoor phenotyping systems from one to the other, or from year to year in the field, how they're actually working. I already mentioned the phase, and Ono will say more about that tomorrow. So we have a phase system, 3 rings, which are providing future CO2 concentrations specifically for breeding. So we will install on this phase system all the phenotyping gears which we need in order to measure in the field. That's the setup which we have in Klein Altendorf. The big advantage which we have at the moment is that we get the CO2 from our big coal burning company, which is RWE. We get it for free. We have these trailers here which are transporting the CO2 from the coal burning plant to the field site. That's pure CO2 from a scrapping facility which they have installed there. That setup is finished. The first full year will be 2015, and a bit more harvest. Volatiles, I also can probably, so this is the patho node. This includes not only the volatiles, which have been shown yesterday, but also confocal laser scanning microscopy unit, which is also installed in Munich, and Yogi can tell you more about this. Is more in the patho background, so this is looking into also labeled pathogens or the effectiveness and the diversity there. It's an automated feeding system, which then is using the confocal as a sensor. In Gadersleben, they have set up a macrophenomic system, also automatic systems to check for pathogen infection, and that's it's a full pipeline now with infected leaves and how you can quantify the infection on there, also the dynamics and the different stages there. In the patho, I think that's, I don't have to mention that anymore. There's a volatile system, which Yogi already introduced yesterday. Where are we? We are in the DPPN project here. This is our setup so far. We had existing infrastructures before, so we are now here. So the next step now, as I mentioned, our DPPN project is coming to the end of the year. So we are now going into the next level. We have the basic funding. We have to establish the basic funding from the institutions, which have been promised during the grant already. Now we are actually going to the next phase, because we are within, most likely this year, we will form a new legal entity, the DPPN Association, which will host the entire continuation of the DPPN project into the next phase, the use phase, and also the 3 organizations, Jülich, Gadersleben, and Jülich, will be participating in there. And we'll have a DPPN Association as a legal entity, which will develop that further to provide access and to also go beyond the duration of the project. Yes, I mentioned all these up here. What I did not mention is access. We are still working on that. We have access specifically for German users. In this specific case, you are actually better off than many German users, because Europeans can apply through year in 2020. There's at the moment no specific call for access for German users on German facilities, but we're working on that. The other thing which I did not mention, because it's a different project, is the integration of bioinformatics, which is very important. There's a national platform for bioinformatics in Germany where Klaus Meyer and Uwe Scholz, Klaus in Jülich, Uwe in Gallerstein, and Björn in Jülich are key persons for the plant side, and therefore we are linking the phenotyping triangle to the bioinformatics triangle in Germany on that.